So let's move ahead to our another speaker, and he is Jonathan Trevor Veneta. Trevor Veneta is a PhD candidate in Minshela Lab at Purdue. When Trevor is not removing parasites from animal organs, he is either outside, wrestling children, or playing Ultimate, sometimes all three simultaneously. So let's welcome Jonathan to show us, after all, how parasites are actually changing the world. Let me share my screen.
Hi everyone, my name is Trevor Veneta, and I'm a PhD student in the Minshela Lab at Purdue University. And today I want to tell you a little bit about what I study, which is how parasites change the world. So the first thing that we're going to talk about is what actually is a parasite. So all types of organisms can be parasites. There's parasitic insects, there's parasitic birds, there's even parasitic plants and mollusks that occur all over the world. And so a parasite is just an organism that lives on or in another organism, and it does harm to that organism. And there's a huge diversity. We think that somewhere between 50 and 80% of the species that exist on Earth are parasitic at some point in their life cycles. So it's sort of this parasites everywhere. For every organism that exists, there's almost always at least 1, 2, 4 parasites that infect that organism. So there's this entire hidden world out there that we're just kind of starting to wrap our heads around how important they might be. And one of the ways that parasites can be important is that they can kill organisms. So this is rinderpest in Africa. It swooped in and it knocked out a massive number of wildlife, of livestock and wildebeest and other things. And it had a huge impact on that entire ecosystem in the Serengeti. But there are other ways that parasites can impact the world, really. And so one way to think about this is what you do when you get sick. So when you get sick, you often avoid other people. You might lay on the couch all day. You might eat different food and things like that. And so animals do the same thing. They might avoid social groups. So they might avoid places where they could get sick. They change what they eat and all different types of things. And so these, although they appear really simple on the surface, can have big impacts on entire ecosystems. And so one example of this is parasites that live in the intestine, these nematode worms that live in the intestines of cows and sheep. When cattle get some nematode worms, and when sheep get nematode worms, it actually increases the amount of methane that they release. So sheep burp out methane and cows fart out methane, and sheep that are infected with nematodes release 33% more methane than uninfected sheep, and they take longer to raise to a harvestable size. So what we have in this situation is where we have methane, which is 25 times more potent than CO2 as a greenhouse gas, is actually being increased by parasites. And so parasites may actually be influencing climate change, which is pretty wild. And so what I do here at Purdue is I've kind of have this idea that, well, animals impact nutrient cycling and ecosystems. All animals get parasites. So how are parasites impacting nutrient cycling? One of the ways that I do this is using math, which we're not really going to worry too much about here. But another thing that I do is I look at infected and uninfected snails, and I look at how much they eat and how much they poop. So in this case, we fed some snails some food. Infected snails up here in the orange, they eat more food than uninfected snails. They poop more than uninfected snails, and they're releasing more parasites than the uninfected snails are releasing eggs. So that's one way that parasites might impact nutrient cycling, is just how much poop is being put out into the world. Another thing that I do is I watch snails crawl around in containers to see what they do when they're attacked by parasites. So this snail here is shaking its shell to shake the parasites off. It's being attacked by parasites right now. And it does this shell shaking, which is really energy expensive. It takes some energy to shake your shell that much and try and get those parasites off. And so that's another way that parasites might impact how much that snail needs to eat and how much it can move around. And the last thing that I do is I actually build ecosystems. So this is a mesocosm tank. I've filled it with snails and parasites, and I measure how many plants there are in there and how much nutrients are in that entire ecosystem. And so that's just a little snippet of what I do for trying to figure out how parasites change the world.
I'll say thank you so much, Jonathan. And so I had no idea actually that all of this goes around in producing greenhouse gases. So I think in the initial two of our presentations, you saw how much it is important for data as well as, assimilating all of them in your lab and understanding and then analyzing. So I'll just check my chat box if we have any questions. Otherwise, we can address all of them at the end. So let me begin.
Yeah, I just wanted to say I've been talking with one of the students, and feel free to send a message. I was replying back privately to Priyanshu, and yeah, but feel free to have a conversation with either one of us of any of your doubts or questions about how to get into university or research related topics.
Yeah, wonderful. So definitely. And I'll make sure that you have everyone's email address, the leads here, the participants here from the graduate program at our NGS website. So at the end of the session, if you want to connect with somebody personally or ask them questions regarding their research, or if it is a general research question, how do I do this? You just post your questions and we'll be as quick in giving your response as we can be. Okay, so let's move on to our third speaker for the day. So she is Christina Schilling-Costello. She is a first-year PhD student in Purdue University's Interdisciplinary Ecological Sciences and Engineering program. She conducts research in Dr. Linda S. Lee's lab and aims to elucidate water quality concerns in Pakistan. Prior to joining Dr. Lee's lab, she was an environmental health specialist in Maricopa County, Arizona. So she obtained a BS, like Bachelor in Science, from the University of Evansville in Chemistry and Environmental Sciences in 2016. Her hobbies are, hiking and soccer. And she has put together a really wonderful insight into her world of analyzing the contaminants in rivers. And she will take you through her field experience, research, and lab work. So I will just very briefly will share. Wait. Let me share the screen.
My name is Maria Cristina Schilling-Costello. I am working on occurrence and fate of contaminants of emerging concern.
So my research focused on contaminants in water from two major rivers in Pakistan. Now, instead of shipping water samples to the US, what we were able to do was to take samples and pass them through a matrix. In this case, it was an HLB matrix in a solid phase extraction cartridge. Now what this matrix does is when you pass water through it, basically it will grab onto the compounds. So with all the compounds in this cartridge, they were then able to ship the cartridge to me. So I was able to extract these compounds from these cartridges under vacuum in this hood. So what happened then is I ended up with these compounds in an organic solvent. In order to analyze even smaller concentrations, what we do is we actually blow down the samples and evaporate the solvent with nitrogen gas.
Samples were then injected into a liquid chromatography system with a specialized mass spectrometer that allows for compound identification and quantification. We targeted quantification of 68 organic compounds. In general, seasonal differences were typically observed, with compound concentrations being low under high flow conditions. High flow conditions resulting from snowmelt tend to dilute chemical concentrations, since these sources are unaffected by anthropogenic activity. Heavy rains during the monsoon seasons could bring in additional chemicals such as herbicides and other chemicals used in agriculture from the soil being eroded into the river. But this may not lead to an observable increase in water concentrations due to the dilution effects from the massive amounts of water. The compounds that we found and the concentrations we found varied at different river locations. A group of endocrine disrupting compounds known as plasticizers called phthalates were found at all locations along both river systems. Bisphenol A, another plasticizer known to disrupt the endocrine system, was detected in most samples but was at lower concentrations than the phthalates. Several pharmaceuticals, including sulfamethoxazole, diclofenac, ibuprofen, acetaminophen, and naproxen, were detected at some locations but consistently at lower concentrations. In the Swat River, total phthalate concentrations range from 100 to 500 parts per trillion during high flow and 500 to 4,300 parts per trillion during low flow. Total pharmaceutical concentrations range from non-detect to 20 parts per trillion during high flow and non-detect to 820 parts per trillion during low flow. Using heat maps, a data visualization tool where the yellows and reds refer to higher concentrations, whereas the blues refer to lower concentrations, the differences between upstream and downstream measurements can be seen, as exemplified here for the data in the Swat River during the low flow river season, where concentrations downstream are higher, as noted by the red and yellow colors, compared relative to upstream, as noted by the blue color. In the Kabul River, total phthalate concentrations range from 200 to 900 parts per trillion during the high flow season, and 400 to 1,000 parts per trillion during the low flow season. The total pharmaceutical concentrations range from non-detect to 10 parts per trillion during the high flow, and 800 parts per trillion during low flow. In the Kabul River, seasonal differences in phthalate concentrations were only observed at a few sampling points. Whereas for pharmaceuticals, seasonal differences were consistently observed, with pharmaceuticals being primarily detected only under low flow conditions. The map showing low flow data for phthalates and pharmaceuticals in the Kabul River did not reveal any clear trend in differences between upstream and downstream, unlike what we observed in the Swat River. The phthalates that were detected in all the river systems were often at higher concentrations than the other organic compounds we targeted for quantitation. Phthalates are known to mimic hormones, thus disrupt the endocrine system that controls sex differentiation in fish, as well as several other health indices. However, based on data from the United States Environmental Protection Agency knowledge base, the phthalates and bisphenol A concentrations in both the Kabul and Swat rivers are below levels expected to produce an observable effect in fish.
Thank you so much, Christina. I think I have stopped sharing this screen. So you saw how in all these three videos, there was a layout for the research. Very first thing, you think about a question that has been bothering you or the society or the environment that we are living in. Then you collect the appropriate data for it. You pick certain tool to analyze those data, and then you see how the trends are, how the variations are, and then you try to conclude what is causing what, and just see that, for example, how Jonathan said, how parasites are changing world. I had never seen that and never heard of that before. I would like to highlight that the videos or the research that we are sharing here is just like pinch of salt, like plethora of research is going on at Purdue, and not only at Purdue, but everywhere across the world. There are just these, highly motivated graduate student who wanted to share their research. So do not limit your thoughts to just what we are showing you up here, but anything. Since you are in your high school, you have, like, the most burning mind. You can come up with certain ideas, certain questions. Don't limit your mind. Think how many questions you can think and then follow it up. Let me see if there is any question for me to address. I think Priyanshu, your question got addressed. Okay. So Angel is saying, I have been telling Priyanshu how to go about getting into research. He is specifically interested in space and astronomy. Oh, I am sorry, Priyanshu. I think we were going to have one person who was going to show how we were getting the signals from the satellites and designing the satellites. We have a very strong astronomy class. I mean, if you know, there are like more than 25 astronauts from Purdue University. So if you're interested, Purdue is the best place to look into it. What is the most important advice for this to maintain your curiosity? As long as you're curious enough about something, you will really enjoy research. I also referred Priyanshu to Purdue Physics and Astronomy website. Yes, absolutely. So surf on the internet correctly, and you'll get a lot of resources, and you have people around here. You can send, if you do not know the email address of the people you would like that to send the email, always send your emails to pgsg.community@gmail.com. And we will find out the best person who can answer your question, and we will, follow up with you. How can I get admission in Purdue University? By applying to Purdue University. Yeah, no, so as I said, you have a long way to go before joining for master's or PhD. You first will have to do your undergrad. And yes, so you'll have to apply to Purdue University. For that, you'll have to keep a good score. You'll have to give some of the exams, GRE and TOEFL. And yes, make a sound statement. Why do you want Purdue University? Why you are motivated about what you want to study? And just apply. There are like plethora. And I'll say there is a strong representative from Indian community also, so you'll not feel that you're not at home. I want to also join. Okay, let's all join. Fine. Also, some of the presenters are here. If you have any question, yes, there are some of the presenters who are presenting in their pre-recorded videos. Some of them are here. So if you have any direct question about their research that we are showcasing here, you can ask those questions too. Okay, so I think you can read the chat box. I will go ahead and showcase you one of our cultural center. So till now you saw mass spectrometer that has been used by to study the contaminants in the river by Christina. And similarly, every researcher uses certain tools. So we showcased you some of the research videos, some of the lab videos. Now I'll be showing you one of our cultural center videos. So how do the graduate students survive here? How do they survive here? I mean, just by having a good community support from Purdue University. And I must tell you, Purdue is the most, student-oriented kind of university I have come across. So yeah, you will enjoy your time over here because we have a lot of things to explore. Let me go ahead and share one of our cultural centers that we have at Purdue.
Todos son bienvenidos, which in English means all are welcome. Not only is that a motto at the Latino Cultural Center, but a guiding principle. As soon as you walk into the Vibrant Center, you will feel that you belong. The LCC serves as a hub for Latinx activities on campus, and a lot of it happens here in the Maricela Alvarado Lounge on the first floor, where students connect with each other through meaningful conversations about Latinx culture and more.
The Latino Cultural Center was founded in 2003. We have been recognized as a national model for Latino cultural centers We are only one of 2 Latino cultural centers in the state of Indiana.
With over 1,000 texts in its collection, the library provides a significant educational resource for Latinx history and culture. You can reserve time in the fully equipped conference room, take advantage of the kitchen facilities for an event, or finish up a paper on the second floor in the university-supported computer lab. The Latino Cultural Center is much more than a building. It's a place to celebrate Latinx culture, a place to find familia, and a place for everyone. Stop by to see what the LCC has to offer.
Okay, wonderful. So you saw, if you have to study or if you want to have fun, you just find your community and hang around with them. Yes, so okay, I think, I'm sorry, my another screen was also playing. So as Hannah is saying, you can also go to Purdue for your undergrad if that interests you. Okay, she did both her undergrad and master's at Purdue. So if you have any questions regarding that, you can definitely ask Hannah about it. And we will list everybody's email at the end of our session. Now, we will move to our next session, that is like the second session. Again and again, I really want to applaud our leads and the participants for making it work. And we have Angel, Hannah, and Sanchez as our lead to conduct the forthcoming sessions. I would like to hand over it to Angel now to conduct the second session.
Hey everyone, it's great to see so many people here, and hopefully you get some nice information out of this event. So my name is Angel Enriquez. I'm a third-year PhD student at Biomedical Engineering. I specifically develop very small devices for medical applications. If this interests you, please feel free to message me in the chat, and I would be happy to answer any questions. So now in the program, we're in session number 2, and I'm going to introduce now Elle Rockefort. And Elle is, excuse me here. Elle is a PhD candidate in sociology, studying social movements and inequality. She's researching full-time thanks to a fellowship, and in her off time, she co-hosts a social science and baking podcast called Proofing and Lies. Oh, that's cool. I'll have to check it out. So Elle is right here with us. I'll ask her to unmute herself. And Elle, can you share screen? If not, I can share the presentation for you.
I think you'll have to do it. I'm not a host.
Okay, then I will share here.
I can make you a co-host. Okay, I'm sorry.
Oh, okay. Please do that. Elle, thank you so much for agreeing to come and be here in person for your presentation. Thank you.
Yeah, no problem.
Right now, I think now you'll have the permission.
Yep. Oh, yeah. Okay.
Awesome.
Hi, my name is Elle Rochford, and I use social media to study politics and inequality in the United States. I'm a PhD candidate in sociology. And my work is looking at Instagram specifically to understand social movements. I'm interested in other social media platforms as well, but we have an intense amount of politics going on in Instagram, despite most of research being done on Twitter and Facebook. Instagram is owned by Facebook, so there are all these interesting connections in the social media world. So I'm gonna pick some of that apart and tell you about how I go about my research. So social media helps us kind of get a window into people and organizations, not necessarily their authentic lives, but the narratives they tell about their lives, about problems, and about solutions. So social media data to me is fascinating because there is so much that is social media. Even things like banking apps like Venmo have social media features. Increasingly, there are exercise equipment that have social networking capabilities. There are even some fridges, right, that are actually social media. So increasingly, social media is a part of our lives and incorporated into all the products we own. So it is a huge boon, basically, as a researcher, because there is so much of it. But it's also sets up a series of problems if you're trying to use social media data for research. So the first thing you want to consider is your research question and what platform or platforms you want to use to analyze it. There are things to think about. Is the platform anonymous? Are users totally anonymous, partially anonymous? Things like Yik Yak, which is now out of favor, was totally anonymous, but linked to your region. Then there are things like Facebook, which are highly identifiable. Even if you're using fake accounts, you're still kind of grouped together with social networks and with photos and names. So you start to think about the features available on each social platform, and that will lead you into more research questions and things like that. Another thing to consider is the platform's reputation. So some platforms have a reputation for being places of information and politics, things like Facebook and Twitter, for example, while other platforms might have a more positive, upbeat reputations, things like Peloton and Instagram, and then others might have a bad reputation, things like 4chan and 8chan, and in its early days, Reddit as well. So you want to select your platform based on your research question. Some questions are going to lead you really nicely into the platform you want to use, while other platforms might be ruled out. And you want to have a working knowledge of how your platform works, whether they update features regularly, things like that. And you're going to want to think of the data you want. Some data you may not be able to access. Capturing stories is problematic unless you're able to go on daily and check stories. So things that maybe have extra security or privacy. It would be hard to do anything with Snapchat, for instance, although Snapchat is falling out of favor. So it's also important to know what social media is popular in the area you're studying, because some platforms might not be widely used. So you want to think about what data you're trying to collect and what platform will be best for that. And you may need to get really creative with that. If you're interested in Venmo transactions, you might have to set things to public and view it on your own, or you may interview people about their Venmo habits and have them show you their screens. So there are ways around privacy settings that are still ethical for research. And all of this involves human interaction, so you would need permissions to catch or collect this data. Don't know what that music is. So my guess is that many of you are on more than one social media platform, especially because so many things are now connected to social media. So I would ask you to think about what platforms are you on? Maybe what platforms have you used and fallen out of use? Do you change the way you interact with a website or the kinds of things you post? A lot of people will separate maybe their Twitter identity from their Instagram identity, from their Facebook identity. So kind of think about how you interact with platforms and what that might tell you, or what a researcher might learn from looking at only one or from comparing across. So who's attracted to a platform is going to be based on the platform design. Being anonymous can often generate more hostile postings. And then there are other things that can promote positive posts. So Instagram, for instance, you can only like react. You can't negative react to a photo. And the poster has a lot of ability to adjust. So they can delete comments. You can actually filter out certain words. So for instance, if you're photoshopping something, you can put a filter. And so anyone commenting about photoshopping, those comments will be deleted. So anonymity and user control will shape the kind of personality of a platform. So once you've decided what platform you're using, you have to think about how you're going to collect that data. There are great coding softwares you can use that will scrape the data for you. But then the other coding, right, not web coding, but coding as analysis, is to systematically organize all the data you have. So this is how we get from information to data to findings. So we take a huge number of posts, for instance, and then we start to sort them into categories to find the themes and the patterns in the data. And there's different strategies, and I've used multiple strategies. You could do line by line coding. So maybe you want to see if people are talking positively or negatively about something. So you might go line by line and count all of the negative words or count all the positive words. And there are softwares now that will do this for you. Thematic coding is a lot more hands-on, so you would have to know about your research subject. So I study political organizations, so I tend to know what their mission statements are and what their key concerns are. So I look through all the posts to see, are they mentioning that? Then how are they mentioning that? And then once you have all this raw information coded into data, you can run that data through different softwares, and you can make regression models, you can chart usages, you can look at trends over time. There's a lot you can do with it once you've gone through the initial coding strategy. So just for a little example of what I do, and this is a very quick example. If you look at the PGSG info page on Instagram, for instance, and just take those first 6 images. So this is a very rudimentary coding. This wouldn't be what I would do for a final project, but you can see some themes immediately emerging. Number one is that the first 6 posts are largely text, right? So we could guess that this is an informational or educational page versus like an Instagram influencer who might be posting products or posting selfies or models or stylized images. So we can guess that this is largely to share information. We see lots of events are listed, which kind of supports the idea that this is about information sharing. We also see a theme of blue, gold, and black. Those are the dominant colors. So that makes sense because Purdue is black and gold. So if we go in blind, we can still pick up on these themes. But if we know that this is related to Purdue and those are Purdue's colors, at least black and gold are, we can start to build theories about what this page is trying to do. So my overall theme that I would say is that this is about support, information, and resources. So when I do my research, it might start something like this, where I would just look at a page and just try to figure out the overall message the page is telling me. And then you go through and you systematically sample, and you can do this with technology, or you can do it by hand, which is the old school way. You can generate random numbers and sample those posts. And then you start to dive very deeply in, and you do multiple rounds of coding. And typically you'll bring in other researchers who can confirm your coding so that you have a sense that your data and your results are going to be reliable, and that someone else could pick up your coding scheme and find the same things with a different sample. So that's a little bit about what I do. I work in other social media platforms as well. And I also do a lot of work that's unrelated to social media. Social media is just the tool I use to study inequality. But you can get really creative with research, and I wanted to kind of put a plug in for social science research, as well as all of the great engineering that's done at Purdue. We have a thriving social science department. Thanks, and I will stop sharing and throw hosting back to, I'm not quite sure who it belongs to.
That's okay. No, thank you so much for that presentation. Very interesting research that's being done over there in the social sciences department. And it's crazy to know how social media can be used in such powerful ways, right? So thank you so much, Elle. And I'll be sharing my screen now because we have this fun game. It's called Kahoot. And what it is, it's it's a quizzing game. So please, everyone, I'm going to send in the chat the link and the pin to join our Kahoot. And we'll be able to have some fun with this. I'll ask some questions pertaining to the different presentations from this session. And if everyone can join in that link, kahoot.it, and then once there, you can enter the PIN, which is 5916517. So I'll give a couple of minutes for everyone to join there. Does anyone have any questions for Elle?
Yeah, I think I have a question. So I understand you look at the data. So what is
I don't
the
think she's here, sorry.
Yeah.
okay, she's not here. So I would like to know, like, what is the quantity of data that she might be looking at when we do the social media analysis? Because coming from a metrological background, I know we can even look at 1 week of data or 10 years of data. But to make any conclusion over there, I was wondering how long data set that they use. But I think I can email her and ask. Let's play the game, Kahoot.it.
All right. I'll give just one more minute, see if more people join. But yeah, the website is kahoot.it, and then we have that pin right there, 5916517.
Come on, join fast. And yeah, because I'm so eager to play the game. Come on.
All right, I'll get to start it. I think you can still join. The link and the pin are still in the chat. So let's get started. All right, 3, 2. All right, so this is the starter question. This is just to get everyone acquainted with the program. And then the second question is with, it pertains to else. Okay, so music, that was the music that was playing. Some people are not ready. All right. Hannah has the quick fingers. All right, so here's the second question. Why is social media research important? All right, seconds. Boom, all of the above. All right, thanks everyone for playing. Let's get on with the second part of our session. Now we are going into Zucro Labs. So let me put the screen to share there. Okay, so this is a video of Sucro Labs. I'll be playing it for a little bit, and let me know if you have any questions after.
Hi everyone, my name is Dana Obanoff, and it's my pleasure to virtually welcome you to the Maurice J. Zucro Labs here at Purdue University. As the largest academic propulsion lab in the world, we here get to do research in combustion, propulsion, explosives, hypersonics, laser diagnostics, and so much more. But rather than tell you all about it, I'll pass it off to some of my colleagues to show you around. And if you have any questions, I'll be online at the end to go ahead and answer them. So sit back and enjoy the tour.
Hi, welcome to ZL1, where there's a lot of research that goes on in terms of laser diagnostics as well as combustion research. Let's go take a look.
As a member of the Meyer Research Group, I work in laser diagnostics and especially combusting flows.
Now what that entails is working on setups such as this, where this is a CAR setup, which stands for Coherent Anti-Stokes Raman Scattering, which allows you to use pulse burst lasers like this big guy right here, which send laser pulses in and can allow for us to measure temperatures in flames without actually disturbing them, because it is a non-intrusive diagnostic. So that's actually very helpful for combustion research, and a lot of that work goes on here in ZL1, in this room, and in another room that we'll just show you now. So this is the inside of a pulse burst laser that we use here at Zucrow Labs. Now, what you do with a pulse burst laser is you take your initial inlet laser and you bounce it through all of these optics and you amplify it using, in this case, neodymium glass rods. And then by dumping a lot of energy into it, you can actually amplify your laser signal, and that allows you to get large amounts of energy compared to what a laser pointer will do, and that's very useful for the techniques that we develop here. So this is another room in ZL1 where we do explosives research with using laser diagnostics. We do the explosions here in this blast chamber, which allows us to do small explosive events. A lot of research we've done lately has been involved with detonators. And then what we've been doing with them is using devices such as this. This is a quad scope. Now the quad scope allows us to image something about here and split the view into 4 different views on the camera chip with this. And using those 4 different views, we can actually 3D reconstruct what we're seeing. So we can end up making evaluations of particle sizes using only a single camera, but 3D reconstructing it, which is actually a very fascinating subject called tone reconstruction.
Hi, I'm Jay Evans. I'm a second-year grad student in aeroastroengineering, and I work at Maurice J. Zucrow Labs, or more specifically, the high-pressure laboratory, which includes ZL3 and ZL8. Just to orient you, ZL3 is where you met Scott Meyer and saw the rocket cell. It houses a lot of the original rocket cell and gas turbine test cell work that went on at Zucrow Labs, and then a couple years ago, construction finished in ZL8, where we have 5 new test cells and an all-new laser lab that spans the width of the building, where students can conduct even more high-pressure experiments and run diagnostics on them. Same kind of work, gas turbine and rocket-related stuff, or even more fundamental combustion, but more test cells and more modern facility space to do it in. All right, now we're walking into ZL3. So in addition to housing the gas turbine and rocket cell, this is also where a lot of the graduate student office space is. So you walk in. It's a pretty nice facility. You have a nice big conference room where you can have the big important meetings. I'll let Ethan kind of pan the camera around in there. And then on the other side, we have professor offices, including Scott, who you met earlier. As we come further along, have a break room where students can eat lunch or interact, get coffee, things like that. Then coming further along, we have our cubicle farm space where students can do various things like CAD or data analysis, anything that would require a computer. There's the workspace to do it in. So moving further along, there are collaboration rooms where various students are officed. Still more cubicle space. And then we start to get to the control room. So here's an example of one. We have a nice 4 monitor set up at each station with large TV screens to, monitor the experiments as they're going on. Here's a bigger version with the rocket cell.
All right, so there's more to the video, but if anyone is very curious on understanding a little bit more about Zucrow Labs, please let me know and I can share this video with them. Now, we are going to head into with another Kahoot. So please bear with me here. This one is going to be on Zucrow Labs on this last video that we just saw. So
And you can still join. I think in the chat box you have the details.
Thanks everyone for answering. Okay, now we are going into our third part of our program. We are going to show you the Black Cultural Center, and they have a video that I will share right now. And here we go.
Founded in a period of Black student activism in the late 1960s, the Black Cultural Center is the most senior of the campus's cultural centers. The first on campus to be designed by an African-American architectural firm, this 18,000 square foot facility, built in 1999, emulates the layout of a traditional African village with a grand circle at the building center. The spirit and richness of the African-American culture can be seen in the artwork, artifacts, and even the receptionist desk on the first floor.
It's actually shaped in the design of the hull of a ship, and the ship has significant historical context for African Americans in that many enslaved Africans were transported from West Africa to the United States in the hull of a ship. One of our hallmark programs is what I call the heartbeat of the Black Cultural Center, which is our performing arts ensembles.
The ensemble groups rehearse and perform in the Antonio and Betty Zamora studio on the first floor. Also on the first floor are a formal lounge and an exhibit hall, which often features student-created work. On the second floor, you'll find an expansive library with over 7,000 books, a large study space, and a university-supported computer lab. For over 50 years, the Black Cultural Center has provided a sense of belonging. We promote academic excellence, cultural pride, and a feeling of home for many students. The center has strengthened the understanding of the African American heritage for the entire campus. We invite all of the Purdue community to visit the BCC.
Okay, so that was the video for the Black Cultural Center, and we have all these.
Hi everyone, my name is Anna, and I work in a.
So we have all these cultural centers across Purdue, and although it's for different diverse communities, it doesn't mean that you have to be from that diverse community to enter. It's more to allow everyone from the Purdue campus to understand the culture of the different centers available. So now we are going towards another Kahoot, and I'll share my screen here. And this one is on the Black Cultural Center. Okay. I think this one was pretty easy. Thanks everyone for playing. All right, now we are going to head into an announcement. I will share here. So there is a summer research internship for high school students that is beginning now for the Weldon School of Biomedical Engineering. This, unfortunately, I think only applies for students in the US, but it's a 9-week-long program with some compensation. And here's the information, the flyer. Please let me know if you have any questions about this summer research internship for high school students, and I'll gladly answer any questions you may have. And let's head to our last video from the program with, excuse me, with Pratik's UV locker. So Pratik is actually here. Maybe you can give an introduction of this research you're going to present.
Yeah, thank you, Angel. Yeah, so as you all know, I'm a graduate student. This is actually not what my research is based in. I'm mentoring a group of undergraduate students. So before you become a graduate student, you'll be an undergraduate student. And the way or the course that I'm mentoring them in is called VIP, right? So VIP, or Vertically Integrated Projects at Purdue University. I'm just putting a link in the chat. So that's one of the courses that you might be taking when you actually attend school over here. And this is an iteration from the summer, so it's a little old. And there's an updated poster of what you've been working on with the same project in the fall. But let me give you a little bit of background before the video plays. It's about 5 minutes. Let me make sure I put, yeah, I think I put everything in the chat. Yeah. So UV Locker, what is UV Locker? So we all know we're in the middle of a pandemic right now. I used to be a teaching assistant for a electrical design course where you had to transfer lab components between different students, and they had to wait like 3 to 4 days to do that. So I was thinking, why can't we just make a locker which disinfects these components? So this is essentially my product idea and how we're actually implementing it through a full product development stage. So if there's anyone out there with a business mindset, this is something that would be pretty cool, and you can actually check this out. I'm gauging that most of the students on this call like the stars and astronomy, but let's get you interested in projects that are on Earth as well, because Earth is a pretty cool place. But yeah, I will let the video do the rest of the talking. So thank you.
All right, man. Thanks for that introduction. Here we go.
Presenting a ultraviolet C germicidal irradiation solution to object disinfection for Purdue campus. This is our team, quite the motley crew of majors in engineering. Thank you everyone for your tireless efforts in making this a reality. A list of contents for this presentation. In order to create a product that would work great with the limiting of spread of COVID-19 in Purdue University, we looked at similar products that are already on the market and compared their functions and materials to help create a more reliable model. With this invention, you're no longer limited to small household items, and it's the first ever remotely controlled UV sterilizer. The need for this product stems from the long disinfection times required to quarantine objects. Here, we provide a secure, safe, and remotely controlled solution to disinfect objects such as lab equipment, documents, keys, phones, etc. in 15 to 30 minutes, as opposed to the standard 2 to 8 days.
Here is a CAD model of the UV locker. On the inside, the UV locker contains UV LED array on both the top and bottom to ensure 100 millijoules per square centimeter irradiation, a transparent shelf, rotating plate, and 4 reflective walls to allow for the UV light to reach all surfaces of the deposited items, as well as a microcontroller, seen in red, to manage the electrical processes of the locker. On the outside, there is an LCD display to display the timer, as well as a keypad for administrative processes. The lock on the door is fully automated to allow for a touch-free experience. Here is the activity diagram highlighting the key steps in the transaction process for a single transaction between Alice, who is depositing an item in the locker, and Bob, who is collecting it.
When you launch the UV Locker app, the user will be directed to the home screen where they log on using their Purdue two-factor authentication. Once the user has logged on, they will be required to choose if they want to deposit an item for decontamination by clicking on the deposit button, or collect the items that have completed their time by clicking on the collect button. After selecting deposit, there are 3 steps for successfully dropping off an item for decontamination. By clicking on the first button, Locker Availability, Alice will be shown a map that has all the UV lockers and their location, and she can select one locker to choose from. After choosing a locker, Alice will have to specify the item type in a drop-down menu when she clicks on the Item Type button. Finally, Alice will enter Bob's username as the receiver and select the deposit button to confirm that this transaction is ready for decontamination. Bob Bob, who is expected to pick up the finished items, will click on the Collect button after logging into the application to track the progress of the items. The locker pickup button will indicate which lockers within that area, including the locker that was selected for deposit, are ready to be collected. The time elapsed button will include the item that has been selected for pickup, the time required for it to be disinfected, and the time remaining until the user can collect the locked up items. The user will have up to 4 days to collect their items, or else an administrator will claim the item using a master key code on the locker's keypad. Lastly, the Collect Items button will prompt a message that states the item in the selected locker is ready and must be unlocked in order to be collected.
As you can see, we are using the STMF0 microcontroller with its power coming from a wall plug that is being converted to a 3.3 volt DC. Then we have USART, which is being used for communication between the ESP32 module and the microcontroller, ensuring that the system is connected to the Wi-Fi. The other peripherals include the 3x4 keypad, which is operational for the administrator only, the LCD display, the solid-state relay that controls the UV LED array emitting UV light, and the solenoid locks that ensure the locking and unlocking of the door. Based off the previous slide, the following are the instrumentation specifications of the product. The total cost of a single prototype is between $90 to $140. A budget of $4,000 with the following breakdown is required for testing and labor charges.
The preliminary design prototype is ready and can be assembled within a week. Testing with UV-sensitive labels and bacteriophage cultures will take 2 weeks. A dry run across 5 campus locations should take 4 weeks, with successful results leading to large-scale replication and a website upgrade to handle the scale demand of a campus-wide deployment. Thanks to my senior design team for the inception of the idea, Brainchild sponsors, and the Brainchild team.
All right. Thank you, Pratik, for sharing that with us. Does anyone have any questions? If not, get ready for another Kahoot.
Like how it jumps onto a product with
I'll try to be the first
500,000 views compared
Yeah.
to the 500 we've had. But yeah, questions, anybody?
Go ahead, you can ask anything. I mean, it'll be beneficial to ask anything related to the what has been displayed over here.
Absolutely.
Wait,
I missed the last part.
ready. Wait, that's why I'm losing.
All right, let's go for the Kahoot once those questions come up. And this is the last one for this session.
Get this one right, huh?
Objects need a tan.
I was telling you, Pratik. Double as
I mean,
a
if
tanning
it's
booth.
large enough, it's pretty small right now. So
it's pretty small. Okay. All right. Hope you had fun with that. Thank you for listening to this session. I'll hand it over to Alka to continue.
Tell me the result of the last one. Did I please this
yeah.
time or
So,
not?
Let me.
It's okay. I'll consider myself.
Our winner is.
Who is? Anna! Congratulations.
All right. Thanks for playing, guys.
Wonderful. Okay, yeah, guys, make sure, I think there will be, or there might be one more Kahoot, so make sure to play. There can be some kind of gift on your way. We'll try our best to do that, especially in COVID times. Yes, so moving ahead with the session, I think you might have enjoyed these two research. I know it's too much to take at this stage of your life, but it's just, peekaboo that this is something that you're going to do in future. So be as open-minded as you can, and let's take a break of 5 minutes. So it's 8:45 here. So we will be joining 8:50 again over here. So just go stretch yourself, eat something, bring something to drink, and we will meet after 5 minutes. In that time, if anyone wants to be in the session and interact with us or ask any question or wants to tell anything about yourself to us, you feel free to do so. I mean, all the participants, so don't shy out. This is the platform that, really, if you can use it well, it'll lead you to somewhere where you have not even imagined about. So use this platform. Our break starts now.
Hey, Anushka, is it okay if we talk through here so everyone can hear like about the different summer programs that Purdue has?
Yeah, I think unmute yourself and just talk. Do you need permission or something, or you can unmute, I believe.
I mean, maybe she doesn't want to speak.
Oh, okay. Bless
But
you.
I can, thank you. But I can speak a little bit about the summer programs. I am really unsure if there are summer programs, excuse me, sorry, for international students. But there are various programs available at Purdue, and I can share some links in the chat box. I was part of SURF, which is an 11-week program where you get matched with a professor and they have an available project for you. And it's very nice, but you have to be an undergrad to join. That's one of the stipulations. So at least for high school students, I think there's few. There's the one that I showed from biomedical engineering, and I can ask if there's the possibility of accepting international students. But the norm is that it's from the United States. But yeah, keep the questions coming. I'll post some links for the summer program, and that way everyone can sort of look into it.
Hi, Angel.
Hey.
Hey, yeah, my name's Spencer. I'm a grad student here too, and I'll be presenting the next session. But I just wanted to kind of piggyback off what you mentioned and kind of regarding the COVID times that we're in right now. We had in our lab an international student who was supposed to come and visit this past summer, but her travel got suspended, of course, because of COVID. But she was still able to work with us, virtually on things, so we were able to still send her data and she was still able to, help us work on projects and get experience. So, I would say if undergrads are interested in it, it's still worth reaching out and seeing if there are those possibilities of still doing something virtually. It's a whole different experience and things, but it's at least something, a possibility, I would say.
Yeah, thanks for sharing that, Spencer. Yeah, that's totally true. Even though right now it's it's virtual, you still can do some good work from the computer, right? So
and build connections and network and all those things.
Yeah, that's
Yeah,
nice.
I agree, and especially because even if we are here, we are working virtually itself. So you just join it and you can work virtually. But how far I know, I think starting February, people are allowed to travel, and people are coming up because a few people who are traveling and will be joining from other countries over here. So I think we will get back to normal kind of situation.
Yeah, definitely now that everything is becoming virtual, I imagine there will be more accepting of those different limitations that there were before for international students. So definitely try. So there I posted summer college for high school students, and it seems that fits for everyone here. So I'll keep posting some more programs.
Yeah, and I think all of these links will also get updated on our website. So feel free to like check that too. Okay, I think we are past 5 minutes, so we should begin the third session. And I'd like to hand it over to Hannah. Come on, Hannah, let's go.
Hi, everyone. My name is Hannah. I'm a second year Master's of Public Health student here at Purdue. We don't actually do research as master's students in the public health program. We can if we choose to, but it's not a requirement. It's a professional program, so it's a little different. Nonetheless, the research is still important in our area. First in our session, though, we have Anna Guttridge. Anna is a PhD student in the Van Ryn lab. She studies addictive substances. She is originally from Ohio, and in her free time, she loves to paint and take care of her many, many plants. So Alka, if you want to share your screen with Anna's video, and Anna is also here if you guys want to ask her a question specifically.
Hi everyone, my name is Anna and I work in a pharmacology lab at Purdue where I test different properties of plants that contain substances known as opioids. The reason that I became interested in this research is because I really love plants and I find it so fascinating that many of the drugs that we use today, such as aspirin, several antibiotics like penicillin, and even some chemotherapy drugs come from plants or fungi. But besides these purified drugs that you can buy or are prescribed, there are many natural products or herbal supplements that are available for people to buy and use. And most of the time, these natural supplements can be helpful to the user, but if the product in question contains addictive substances such as opioids, problems may arise such as drug abuse. So again, in my research, I study the opioids that are present in these natural products and determine whether they're promising or problematic. So I'm sure that many of you have heard of opioids, such as morphine or fentanyl, which both have contributed to the infamous opioid epidemic in America. These opioids bind to opioid receptors, of which there are three, the mu, the delta, and the kappa opioid receptors. Many of the opioids you've probably heard of, like morphine and fentanyl and oxycodone, these all bind to the mu opioid receptor to create their pain-relieving and addictive effects. When the opioids bind to the opioid receptor, they trigger different signaling pathways inside of the cell, where the receptor can signal through G proteins, leading to a decrease in cyclic AMP in the cell, which is, which is a very common messaging molecule inside of the cell, or the receptor, through a few extra steps, can also recruit another protein called beta-arrestin to lead to other different signaling pathways downstream. So you see that when an opioid binds to an opioid receptor, there's a few different things that can happen inside of the cell. Well, it gets even more interesting because recent research has indicated that different opioid structures can create unique signaling pathways after activating the opioid receptor. For instance, one opioid structure can force the receptor to signal only through the G protein pathway, or maybe the opposite. Maybe it forces the receptor to signal only through beta-arrestin. Of course, it could also be an unbalanced combination of the two of these signaling pathways. But regardless, think of the signaling pattern as a unique fingerprint to that opioid. And this unique fingerprint can help us find new drugs to treat pain relief and alcohol addiction with fewer side effects that led to the widespread opioid epidemic. And so knowing this, I study different natural products that contain opioids to determine what their unique opioid signaling fingerprint looks like and whether the signaling profile contributes to negative side effects. The two plants that I study specifically are kratom, which is from Southeast Asia, And many people use this to self-treat drug addiction. And akwama, which are seeds from a tree in West Africa that people have used historically to treat pain. And so in my research with Kratom, I've used cellular studies to determine that unique opioid signaling profile. And then I take those opioids and I look at them in mice. For Kratom, I used mouse models of alcohol addiction. And we actually found that the opioids that are in Kratom can decrease alcohol drinking through their signaling at the delta opioid receptor specifically. In my research with akwama, I again used cellular experiments to look at that unique signaling profile of those akwama opioids. And then I tested them in mice to see if they did have, or if they did provide the pain relief that historically humans have used it for. And we found that although akwama does have opioids that bind to the mu opioid receptor, remember that's the same receptor where morphine binds, the akwama opioids aren't potent or strong enough to decrease pain in the mice, unfortunately. So to wrap up, my research on both akwama and Kratom helps to better characterize the properties of the opioids that are present in these plants, and may provide guidance for their future regulation. Again, my name is Anna, and if you guys are interested in hearing more about my research or have any questions about research in general or higher education, please feel free to reach out. I would be happy to talk. Thanks.
Thanks, Anna. If anyone has any questions, feel free to put it in the chat. Like I said, Anna is here right now if you're wanting something specifically answered about her research.
I don't know, but I'm curious. I wanted to ask one question. Anna, so I understand that you work in Van Ryn lab. So what does this lab is about? I mean, what all goes around? Because I remember when I was studying sediments in rivers, at that time there was a classical paper from Van Ryn. So I thought there is only Van Ryn because only one Van Ryn, but it seems like there is something to do with it, biology also. So can you please highlight about what your lab works in and what all you do there?
Sure, yeah, great question. Really, the Van Ryn lab, we study pretty much anything to do with opioid receptors. So specifically my research, I'd look at like biased signaling at the opioid receptor, which is that unique fingerprint I was talking about. But a lot of other research happens based on like seizures that happen with opioid receptors. There's a lot of research about like modeling opioid receptors in computers, you know, to figure out which drugs can bind to those receptors. And we're recently starting to move into cardiac research, and that's because some opioid receptors are expressed in the heart. But I would say that the most part of our research is about neuroscience and opioids in the brain.
That's interesting. So do you also use any coding language or something to do your analysis, or you have like some softwares to do that?
I don't personally in my research, my research is more behavioral, but there is a new student in our lab who is starting to get into a lot of computational modeling.
Okay, so you can do like both computational as well as the observational thing.
Yeah, yeah. Mm-hmm.
Okay, wonderful. Thank you, Anna.
You're welcome.
All right, we'll go ahead and move on to our next presenter. But like I said, you guys can throw questions in the chat at any time. So our next presenter is Riley Ratcliffe. Riley is a PhD student in the Hoverman Lab. He is from Utah, where he was raised by mountains and deserts. Even after 4 years in Indiana, he is still constantly amazed by the height of the trees and the abundance of water here. His ideal Saturday involves being deep on a trail en route to some natural wonder. And I wonder how many of those are actually here in Indiana, but hopefully enough to keep him occupied. So Alka, if you want to go ahead and start Riley's video.
But
And we'll
just
watch his-
before that, I'll say, for some reason, his video has a little bit less volume, so you can increase your computer volume if you want.
Hi, I'm Riley Ratcliffe, a graduate student in the Hooverman Lab in the Department of Forestry and Natural Resources at Purdue University. Like I use pesticides around my home to protect against insects that might damage my house or my yard, or even bite me or my family. I also depend on the pesticides used on fields all over Indiana to grow the food that keeps us all going. Some of these insecticides inevitably end up washing off our fields and homes and into the ditches and gutters during rainstorms. These, in turn, feed into our lakes and creeks where they can have unintended consequences on harmless aquatic insects. My research focuses on how commonly used pesticides impact sensitive aquatic insects. Aquatic insects play an important role in decomposing dead things like leaves that fall into the water this time of year, as the insects break down and eat plants. They in turn become food for larger organisms like fish, salamanders, and even birds. Too much pesticide in the creek can kill the most sensitive aquatic insects like mayflies and caddisflies. Even if the pesticide is too low to kill them immediately, it can weaken them and increase the rate at which predators eat them. To keep pesticides out of creeks, always follow the instructions on the container. Don't spray near water or ditches, and don't dump leftovers in the gutter, where they will wash into the creek. Healthy aquatic insects are essential to maintain a healthy stream ecosystem, which keeps our water clean and our environment healthy. That's good news for fish, good news for aquatic insects, and good news for all of us.
Thanks, Riley. And Riley is with us as well. If anyone has specific questions for him, I'll wait just a minute to see if anyone has any.
That was really interesting, Daniel. As I said, like, this was too close to the, insects and all. And some people who do not get along with the insects, it is like, oh, what are you showing? Are you like completely cool with these insects, or you had those, nervousness when you started your experiments, especially with these insects?
I've had a lot of practice with them, so like I've handled them a fair bit, and like mayflies don't bite or anything, so they're just like those little tiny ones, and they just crawl around. So they're actually really
just
cool.
that crawling feel like, oh, what is it?
They're really fun, and they're just like tickling, so it's good.
Yeah, you're fond of them now.
But I know there are a lot of people that like spiders. A lot of people have issues with spiders, which is weird because they're tiny. Like you can squish them in your fingers, and no big deal.
Especially that spring event which happens, they just spit the bugs. I always just wonder, how do you do that? Interesting.
Cricket spitting contest. Yeah.
So there's a question in the chat from Priyanchu. He asks, there's any chance that some insects might have adapted to the chemicals used in pesticides, or maybe in the future?
Absolutely. Actually, that's one of the things that we're researching right now, is looking at differences in pesticide tolerance across different populations of insects. And we're writing a paper on it right now. Like that's what I was doing yesterday. So there's definitely evidence of it happening in like pest insects that eat crops. There's lots of resistance in crop insects. So we're trying to find it in non-pest insects. Mayflies don't eat crops. Nobody's really worried about them attacking fields, but they're developing tolerance as well. So definitely happening. So another question there, how to use pesticides effectively or in a useful way? Well, I mean, they're highly effective, but if we build up resistance to them, then they'll become less effective over time. You've heard about like super microbes, the idea of like a bacteria or something, or a germ that builds up resistance to antibiotics or something like that. The same thing happens with insects. And so the trick, just like with antibiotics, is to switch which kinds of insecticides you're using so that they don't like only see one kind and build up resistance to that. And to use as little as possible. If you use too much of it, then you're going to drive the resistance even faster. So if you can use less insecticides, change which kinds you're using, and any way to avoid using it at all, like if you can plant at a different time or introduce natural predators that would keep the insects down that way. Anything that you can do to use less insecticide will reduce that resistance potential. Alternations to their DNA. Oh, that's a good question. There are people working on mosquitoes. That's one of the big things we used insecticides for, is to control mosquito populations because they're vectors for diseases like malaria and so forth. And there are people that are trying to alter the DNA of mosquitoes so that they can put little death genes into the population and control the populations that way. And I think I've heard that this summer they got approval to do a large-scale test in Florida on that. So not sure how it went, but people are pretty nervous about that because there's something about making a genetically modified mosquito and then releasing it into the wild that's kind of like, oh, well, what's going to happen? And nobody really knows, but we're poking at it. So we'll see.
That's super interesting. Yeah.
Yeah, very interesting. I know my area of study looks a lot at the antimicrobial resistance for bacteria, and it's interesting to see how the two kind of go together and are very similar. If you guys have other questions, feel free to throw them in the chat. I'm sure Riley will stick around for a bit to try and answer those. But we'll go ahead and move on to our next video. It's another one of the cultural centers here at Purdue. It's the ARC, otherwise known as Asian American Asian Resource Center. So Alka, if you want to go ahead and start sharing that video.
Welcome to the Asian American Asian Resource and Cultural Center, also known as the ARC for short. The ARC was founded in spring 2015 as a result of more than a decade-long activism by Purdue students supported by faculty and staff. The first floor features a lounge, the central gathering place where students can relax, hang out, participate in events, and discover the diversity of the Asian and Asian American communities on campus.
So Purdue University is home to more than 10,000 Asian American and Asian international students. The ARC's mission is to weave the diverse experiences of Asian American and Asian international communities into the fabric of Purdue's campus life.
The center features a growing library of books, movies, and games for checkout. It also features spaces for student organizations to hold meetings. You can get a coffee or grab a snack from the kitchen before heading upstairs to study in the university-supported computer lab. The ARC holds speaker and conversation series events throughout the year that examines various Asian and Asian American identities and diversity. The center also invites Purdue's campus to cultural programming to engage the rich Asian cultural heritage, arts, performances, ceremonies, and celebrations. In many ways, the Asian American and Asian Resource and Cultural Center is a home away from home for the growing Asian population on campus. But more importantly, it's a place for everyone. We invite you to stop in and check it out.
Yeah, so like a lot of the other resource centers, it's a great place just to find a home for students to have a place to go study or even eat lunch, get some coffee, whatever they need to do. So our next presenter is Spencer Siddons. Spencer is a PhD candidate in the Surly Lab. Yeah, you can correct me, Spencer, if that was wrong. Spencer's lifelong obsession in all things amphibians guided his research interest to help conserve frogs and developed his interest in scientific illustrations and wildlife photography. When he's not looking for frogs, Spencer enjoys rock climbing, camping, and cooking. And Spencer actually has 2 videos, one that highlights his research and then one that highlights the lab. So we'll go ahead and play both of those. And then Spencer is also here like the other 2 to answer any questions you guys have.
Yep, so I'll share the screen and use this opportunity. He's present here, so you can ask him as many questions related to toads and frogs as you can.
I'm Spencer Siddons. I'm a PhD student in the Department of Biological Sciences at Purdue University, and I'm here at the Ross Biological Reserve to talk to you about a disease infecting amphibians throughout the world, including right here in Indiana. This disease I'm referring to is chytridiomycosis, caused by an archaic chytrid fungus that I will refer to as BD. This is one of many different factors that are contributing to global amphibian declines that we've been seeing in recent decades, leaving 33% of amphibian species threatened with extinction right now. Now, BD is a fungus that has a motile zoospore stage, so the spore swims through the water, infects the frog's skin, ultimately clogging their ability to drink and breathe through their skin. Now, a lot of reports have found BD in infecting frogs in Indiana, but what has been left understudied right now is how BD survives throughout the winter. And so I'm interested in studying how BD infection varies across species of different overwintering strategies, as some will overwinter in the water, some will overwinter inland. And this will help to pinpoint which species are harboring BD infection throughout the year. And this is increasingly important as winters shorten and temperatures continue to rise as a result of climate change. And this is important for 2 reasons. It can help to direct management efforts on those species that are harboring infection throughout the year, and can help to predict future chytridiomycosis outbreaks in these regions, ultimately helping to conserve amphibian populations in Indiana and in other parts of the world.
So this was his research video. I'll just share his lab experience. But before that, give me a second.
Hey everyone, I'm here at my lab at Purdue University taking care of some of our tadpoles that we're going to be using for an experiment here in a couple weeks. They're right behind me, so let's go ahead and take a look at them. These are green frog tadpoles that I collected as eggs just a couple months ago from a nearby site, and we'll go to the container, and I got all these containers filled with them, but this one has the most, and we'll take a look at them. There are all these little brown specks that are swimming around. And you can see how small they are, and even though I collected them a couple months ago, they're still pretty small. And that's because these develop pretty slowly, so these will actually not become frogs until next year. So they overwinter as tadpoles. They stay underneath the ice, they slow down because the temperature is really, really cold, and that slows down their development. So they actually won't be frogs until next summer. And you can see the little brown specks. It kind of looks like dirt almost at the bottom here. And that's actually their food. So what we feed them is actually rabbit food. And these little pellets of rabbit food are filled with plant material, they're filled with protein, help the tadpoles grow up really nice and strong. I pulled out a couple here so we can get a slightly closer look at them. There's kind of a big one at the bottom, kind of a slightly smaller one at the top, and these are from the same parents, okay? So these are from the same egg mass.
And we can see there's a lot of variation, even though they hatched at about the same time, there's a lot of variation in their size. And that might be due to competition. So one of those individuals might be able to eat more, eat faster. It also might be due to their genetic variation. So some individuals might have their genes or their DNA might help them to, or make them be a little bit larger, make them be a little bit longer, kind of like how we have variation with our siblings and how tall we are. Some of our siblings might be taller, some might be a little bit shorter, and there's a lot of variation there. So yeah, so these tadpoles are going to be used for an experiment where we expose them to road salts, like the salt that's put on roads to de-ice them during the winter time to make it safer for us to drive on. And we're also going to expose the tadpoles to a disease. And we're going to see how the disease and the salts impact the survival, impact the growth, impact the feeding, those kind of things of the tadpoles. And then we'll be better able to help manage our use of those salts and how that impacts diseases in the environment and eventually how it impacts the frogs in the environment. So we'll see how that goes. We have to do a lot with the pathogen. So we're gonna culture up the pathogen and we're gonna expose the tadpoles to them in a couple weeks. And then a little while later, we're gonna finish up the experiment and see what we found.
Awesome. Thank you, Spencer. I see there is a question here in the chat. What are the biggest challenges you face when working with frogs?
Yeah, that's a great question. Well, in my research, I look for frogs a lot of times, most of the time in the winter. And that's the time when it's the most difficult to find frogs because they're essentially hiding from the freezing temperatures. And so they don't want to be found. They're underneath the water, underneath the ice, they're underneath the ground. And so it takes a lot to just initially find them. And so we've had to, employ a few kind of newer strategies where we kind of shovel or hammer through ice in lakes and then set traps. So we have like fish traps, small fish traps, and we just put them underneath the water and then come back the next day and hope that we find some there. So that's a fairly big challenge.
I actually had a question with the tadpoles. You said you feed them rabbit food. Is that just, something that's easy to buy that's kind of the closest to their normal dietary settings?
Yeah. So that's one of the reasons. Yep. So it overlaps a lot with their natural diet. And it is one of kind of several food items that we can feed them. So sometimes we just feed them fish food as well. Just the normal fish flakes that you feed your pet fish. There are also algae pellets. So a lot of tadpoles will eat in their natural environment. They'll eat algae that's growing on like the side of sticks and logs and rocks and things in the water. And they'll just kind of like scrub away at the algae that's attached to those things. And so we have little pellets of algae that we can feed them too. Yeah. Okay, another question. What do you do with the frogs sample when experiments are done? Do you just kill them? So when we do have experiments, we do have to euthanize them. It's very sad. It's a very sad day, but we can't put them back into the wild because they may have acclimated to the lab environment. So they wouldn't be as strong or as capable of kind of surviving out in the natural environment. Also, there are certain pathogens or parasites that they might pick up in the lab that we have that we could then transmit over into the natural environment. So to do this, we need a lot of permission from the university, and we have to justify the species that we're using, the number of species that we're using, to control how many we're taking from the wild to use for our experiments. We also need to get permission from the state of Indiana to do this as well, so they can monitor, who's taking the animals from the wild and how we use them. Yeah, great question. And then when there's another question. What do you do with the frogs that you just kill them? What variables do you use to measure their efficiency under certain climatic conditions or under certain dietary conditions? So if I understand this right, under certain climatic conditions. So some of the things that we do when looking at certain climatic conditions, like winter conditions, is to look at their, and this kind of falls under the dietary conditions as well, is we look at their growth. So we look at to see, we measure, when the experiment starts, we measure their size. We measure how much they weigh and their length. And tadpoles can also be measured by their developmental stage. So initially when they come out of their egg, they essentially have no limbs. But as their limbs start to develop, that is a measure of how old they are, essentially, and how close they are to metamorphosing or developing into frogs and hopping out of the water. So we measure all of those things at the start of the experiment, and we measure those things at the end of the experiment. And that can help us to track how fast they're growing, how fast they're developing. And those kind of things are really important to measure, because a larger individual at metamorphosis, so a larger frog when it first hops out of the water after it's been a tadpole, has a higher chance of surviving through adulthood and to then reproduce. So if we see a lot of frogs that we expose to road salts are really, really small, that might suggest that those populations that have road salt exposure may not grow or may not survive long enough to reproduce, to put more tadpoles back into the population, and then those populations could decline. Yeah, so great question. And I have, if there are other questions that pop up, that's fine, but I have an assistant here, and I'll see if she cooperates here. But I have a pet frog here, and this is one from an experiment that I did a year ago, and this one we didn't actually need to use, but so I adopted it. So I didn't have to euthanize it. I just couldn't do it. I mean, look how sweet she is, and she might get a little anxious. So I have a little worm here, and we'll see if she takes this. Oh, there she goes. Oh yeah, she's hungry. So this is a great tree frog. They're found here in central Indiana and all throughout like the central and eastern US. And there she goes. Oh, she loves them. And these actually survive winter. She's not cooperating to look at you all. These survive winter by freezing over 50% of their body. So they don't stay underwater. I'm just checking you guys out. They don't stay underwater. They don't burrow down into the soil. They actually produce sugars, a lot of sugars in their body that helps protect their cells. And then all of the space around their cells freeze. So their heart stops, their metabolism stops, everything stops. And then in the springtime, when the snow thaws and everything thaws, they go and hop around. So what I did with some of these is to see if their freezing ability was the same if they were exposed to that disease that I was talking about, that chytrid fungus. And if they were able to produce enough of those sugars to allow them to freeze throughout the winter compared to if they were not exposed to that fungus. Because like I said, it's really unknown how the fungus operates and infects and impacts frogs in the wintertime. So I have all of those samples, and I'm currently looking through them now and hoping to see some cool results.
This is really interesting. I remember once I was, changing the soil of one of my pots plant. And, that plant was there since, I don't know, for last 5 years or so.
Yeah.
unaware. I was not wearing gloves, and I was just figuring out that I need more soil to fill in because the plant was growing big, and it is one of those kind, succulents kind.
Yeah.
And I suddenly touched something is softer inside. I was like, what is it? And suddenly a frog popped out.
Oh, no way.
Yeah, I was so amazed. I was like, since when he is here? I have no idea. Can they like remain inside soil, that too in a pot inside a lab where we don't study frogs? Can they stay for like last 2, 3? And I'm not where it jumped in, from where it came, I have no idea.
Yeah, I have come under the idea that frogs can be anywhere. I feel like they are very, very, or a lot of species are very, very tolerant to very strange environments. And like tree frogs, for example, they will actually, instead of burrowing in the tree or crawling into the tree holes or like the loose tree bark, which they do in the forest, they'll crawl into the siding of houses or the attics of houses or things instead.
Yeah.
So they've really adapted to a lot of different environments. And then when they're in those houses, they go around streetlights where all the bugs and all of their prey items are attracted by the lights.
Yeah.
So they love streetlights and house lights and things. And so to be in a pot of soil, that's probably surprising to see right away, but they have a little bit of soil. If there's good moisture there, if there's some food that's around there, then they're okay. Yeah.
I completely agree, because even at my home, even once in a while, I just see a frog. I live in the second floor, so I'm like, where did it come from? And the color is similar to my rug's color, so you can identify there is a frog out there. But now I understand. She's adorable, by the way. She's also looking at us. Hi. Wonderful.
Sanchez, are you a fan of this or no?
Nah, not really.
No?
I think it's pretty interesting, the project, but man, I think
well,
not really a big fan.
If you ever want to check them out a little bit closer, or or go look at them in the wild or anything or whatever, let me know.
All right, thank you. Appreciate it.
Yeah, more you explore anything, more interested you become.
Yes.
Yeah, well, thanks, Spencer. That's awesome. I had no idea you're going to have a frog for us. So that's really cool. I'm sure, like the others, Spencer will be good to stick around for a few more minutes if you guys have other questions that you want to put in the chat. We do have one more cultural center video for this session. This one is the Native American Educational and Cultural Center. So Alka, if you want to go ahead and start sharing that video.
Bonjour, aye, aloha, and yat-e. This is just a small sample of greetings from the 65 plus indigenous nations represented on Purdue's campus. At the Native American Educational and Cultural Center, your heritage has a home. And you immediately feel that sentiment as you enter the lounge area, where students socialize, gather for meetings, and share experiences surrounded by historical and contemporary artwork representing a variety of indigenous communities.
The NACC provides a safe and inclusive learning environment for Purdue students, faculty, and staff to engage in meaningful conversations regarding Native American, Alaska Native, and Native Hawaiian people. We proudly serve the campus community in providing a variety of programs and services throughout the year, and one of our signature programs is the Sloan Indigenous Graduate Partnership Program. This is designed to bring Native American, Alaska Native, and Native Hawaiian students who are pursuing a master's or PhD in a STEM discipline.
You can discuss tribal nations in the multi-purpose room, grab a coffee from the kitchen, or hold a meeting in the fully equipped conference room. More educational opportunities can be found in the second floor library and university-supported computer lab. The Native American Educational Cultural Center is not only a home to Purdue's intertribal communities, but a place for everyone to explore. Please visit us to learn more about the many indigenous cultures that are a part of Purdue's rich diversity.
Yeah, so like the others, it again is just, a place for students to go, celebrate their culture, and then also a place to hang out, study, things like that. That was the last video for this third session. Again, you guys can always throw questions in the chat. We'll be watching that. But I will go ahead and hand it over to Sanchez for our next session, unless Alka has any comments in between.
Yeah, no, I just want to thank all of you once again. I can't thank enough. And thank you to all the students who have participated and have stick around till now. This is our final session, the fourth session, and we have 3 research videos and 1 cultural center video over there. And honestly, for me, this is the most educational Saturday I have ever been in. So I think you guys are also enjoying, and feel free to, chime in, just unmute yourself, ask, or even if you don't ask, don't want to ask, just give any of your insight or whatever the research or any question that is coming up to you. So to all the students, feel free, interact with us more. You won't get this opportunity every other Saturday, So yeah. Yes, Sanchez, over to you.
All right, thank you so much, Alka. Good morning, everyone. Thank you for sticking by, and welcome to the last session of this event. So my name is Sanchez Filopes. I'm a first-year master's student in agriculture economics, and it's really a pleasure for me to be presenting the last session of this event. And guys, a lot of us, we've been through that process, that critical process in which you kind of figuring out what you want to do, what you want to do with your life, figuring out what you're really interested about. And sometimes people underestimate how tough it is to really know yourself and know what you want to do, what you want to study. And yeah, do not hesitate to ask any question that you have. There's no stupid questions. Any questions about the process, about grad school, about college, anything that you want to know, please, we're here to help you. Okay, so first of all, for this session, I think it's going to be a really interesting session because we have 3 different presentations, and we have 1 cultural center video. So we will start first with Claire Shred. Claire, I think that I hope that I have it right. I have this right. Claire Shred is a defended master student in the Christie's lab. When she's not wrestling fish, she enjoys rock climbing, and she will be presenting about population genetics and fisheries management. So Alka, I don't know if you can please go ahead and share Claire presentation video.
So, hi everyone, and thank you for joining me. My name is Claire Schreit, and I just finished up my master's degree here at Purdue. And the lab I work in works both in the Department of Forestry and Natural Resources and the Department of Ecology and Evolutionary Biology. And we use genetics to think about how fish in the Great Lakes should be managed. And I think this is pretty cool because in the summer, I get to spend my time out on boats on Lake Michigan, as you can see in this picture, catching some really cool fish. And in the winter, I get to do some fun laboratory work and a little bit of bioinformatics. So let's get started. So who can spot the differences between these fish? Are there any differences? Maybe size, color, number of fish? So this is actually a trick question. On the surface, there really isn't any difference between any of these fish. And this is actually a real world challenge that fisheries managers face when they're trying to identify populations. Particularly for fish species, we can't observe them in their environment, so we really don't know who's related to one another or how they're interacting with each other. And so what if I told you that there are 3 distinct populations here, and you needed to manage each one in a different way? How would you decide to group them just based on this photo? So let's put down some lines. Maybe we can try organizing these units spatially. So let's make the top right maybe 1 management unit, And maybe let's just split that bottom chunk into 2. Okay, so now that we have our management units drawn, let's see how we did. Oh, so we didn't do a really good job at this, but how are we supposed to do better when all the populations look like this? So luckily, using genetics, we can figure out how fish are related to each other based off their DNA. And you guys can sort of think of this as 23andMe, but for fish. And using genetics, we can start to parse apart what those populations actually look like, despite the fact that fish might look the same on the surface, just with the naked eye. And I use this method to figure out how populations of yellow perch are structured in Lake Michigan. So in this top left corner, you see a yellow perch. And they're a popular fish species for recreational fishermen, and they're still commercially fished in some parts of the Great Lakes. However, this population crashed in the late 80s, and despite our best management efforts, as you can see here, even through 2015 through now, these populations are still not doing very well. And in order to make sure we're doing the best possible job of managing yellow perch, we want to delineate populations in Lake Michigan and make sure we really know what we're trying to manage. And we accomplished this using a couple of steps. So step 1, we collected Lake Michigan individuals from all around Lake Michigan. Step 2, we used genetic information from these individuals to figure out how they're related to one another. And step 3, profit. Just kidding. What we found is that yellow perch and green bay are genetically distinct from the rest of Lake Michigan, and they'll probably need a specific management plan. Future work on this project will include trying to further differentiate population differences among fish in the main basin, but that's going to be someone else's research project. Other people in my lab look at different species in the Great Lakes, including Atlantic salmon, pink salmon, and sea lamprey. And we really have a good time out in the field collecting specimens, extracting DNA in the lab, and of course, using our research to inform real-world management decisions. So thanks guys so much for listening to my quick little talk, and I really encourage you to feel free to drop me a question and let me know what you think. Thanks.
All right, thank you. But unfortunately, I don't think Claire is here with us, but we do have a Kahoot questions. Alex, can you please confirm if we do have Kahoot for this presentation?
We do, yeah. I will get that started right now.
Yep.
And I'll request everyone just join it. It'll be fun. And as I said, you never know, the winner can get something. Even if you are, off seas, we can always ship it to you. I'm not promising though. So this 4740122 is the pin, right?
Yep, exactly. I will share it in the chat just so everyone has it. So I'll just wait like a few more seconds just to make sure everyone has a chance to get on. And then we can go ahead and start. Okay, I will go ahead and hit start. You can still join later, so you'll still be able to join later if you're interested. All right, so that was it for just the first presentation, but we'll have more questions for the other presentations.
Yeah, most definitely. Let me just go ahead. With the second presentation, we'll have Kauthik Srinivasan, and Kauthik is here with us. So we will show the video first, and then you'll have Kauthik available to respond any questions that you may have, guys. So just a brief summary about Kauthik. She's a fourth year PhD candidate in the lab of Dr. Nicolas Neunedge. His research focus is trying to find 3D structure of an essential protein complex found in the outer chloroplast membrane of all plants. He's from Delhi, India, and likes to balance work with jogging and growling in a metal band. So Alka, please, can you share Kauthik presentation, please?
Welcome, Karthik. And as Sanchit said, Karthik is available. So make use of it and ask whatever question you have to him. I'm going to play his video, and it's awesome.
Structural biology is looking at life in 3D. All biological processes, when looked at on the microscopic level, involve an interplay between DNA, RNA, and proteins. To find the 3D structure of these biomolecules is to discover the relative orientation of their atoms, which in turn gives us an idea of how they function. At Purdue University, the Hochmeyer Hall of Structural Biology is well equipped to conduct X-ray crystallography and cryo-electron microscopy, two key techniques that can reveal the atomic structure of biomolecules. Let's look at how we do it. It all starts with the identification of a target molecule. This is the BAM complex. It is an outer membrane protein complex consisting of 5 proteins, BAM A through E. This complex is present in all gram-negative bacteria, which include pathogens like Neisseria gonorrhea, which cause deadly disease. Discovering the 3D structure of the BAM complex can help us design drugs that can bind to it, block it, thereby killing the pathogen. Structure-based drug design is one of the many applications of structural biology. The first step in revealing the structure requires the DNA sequence of a protein. This is followed by molecular cloning, where we insert this piece of DNA into a bacterial E. coli cell. We then produce these cells in nutrient-rich media and induce them to produce the protein of interest. After harvesting the cells, we lyse them, cracking the cells open under high pressure to extract the protein they have produced. This lysate is then taken through many purification steps involving techniques like chromatography to finally acquire purified protein. The purity of this protein can be checked by running them on SDS Page gel. Each band you see here is a component of the BAM complex. Now that our protein sample is ready, we can do structural analysis on it. To reveal the structure using X-ray crystallography, one needs to first crystallize the protein. This is done by chemical screening and plates such as these. We use robots like the mosquito and the dragonfly to automate the chemical screening process. This crystal hotel stores these plates and images them at regular intervals to check if a condition becomes suitable for crystal growth. Once we identify a protein crystal, we loop it under a microscope and freeze it in liquid nitrogen, which is at a negative 320 degrees Fahrenheit. The crystal is then mounted in an X-ray machine like this. A rotating copper anode generates the X-rays, which are guided towards the crystal. Once they hit the crystals, the X-rays get scattered and are captured at the detector. The output is called a diffraction pattern that looks like this. Using computational tools, we can use this diffraction pattern to acquire the 3D structure of the protein. Another emerging technique for determining protein structures is cryo-electron microscopy, or cryo-EM for short. Here, we do not need to crystallize our protein of interest, but instead dispense our protein onto tiny grids. We can then either stain the grid for negative stain electron microscopy, or plunge freeze it using this instrument to prepare the sample for cryo-EM. This is the Titan Cryos, a state-of-the-art electron microscope. Grids are mounted here after being frozen. The electron microscope then images these grids to acquire 2D views of our protein in all different orientations. Using computational software, we can use thousands of these images to reconstruct a three-dimensional view of our protein of interest. Discovering the structure of a protein is just the beginning of many more exciting paths. This structural information can be used to create realistic movies called molecular dynamics simulations, and the protein can be mutated to determine the mechanism of action. Drugs can be designed to bind proteins and modify their function. These drugs can cure diseases, unlocking the full potential of medical applications. The future of structural biology looks bright, and the large number of Nobel Prizes awarded in this field is a testament to that fact.
Right. Thank you, Kafrik. I don't know if there is any question in the chat for Kafrik. Nope. All right. So if there's no questions, we will go.
Actually, I
Go
do
ahead.
have a question. Yeah. So this was really interesting. First of all, like because I come from an engineering background, so we only look at or generate hurricane tracks and look at hurricanes and how much rainfall is it bringing to urban landscape and things like that. So, but seeing all of this going around in biological and drugs industry, uh, Karthik, can you please highlight on what's going on right now, especially with the COVID? I mean, I know we have some kind of injection that is coming up, but why is it hard to, come up with a drug that can cure this?
Can you hear me properly?
I can, yeah.
Good morning, everyone, and good evening to people in India. The video might have been a little overwhelming. We made it in a very short time, so
This was huge. If it was in the
yeah,
short
it's huge.
time, this was wonderful.
But so I also have an engineering background. I did my bachelor's in mechanical engineering.
I don't know. There is a music coming up from somewhere. It can be me. Yeah, actually, it was me. I'm sorry. The follow-up. Yeah, please go ahead, Karthik. I'm sorry.
So yeah, as we know, like as the video just showed in a brief glimpse, structural biology can be used to find the structure of biomolecules. When it comes to viruses, they're actually pretty easy to solve the structure of because they're really big. And the smaller the molecule, the more difficult it is to find its structure. With viruses, however, the problem is that they have a very high mutation rate. And so even though structural biology is being used right now to sort of find like the 3D structure of the proteins of the crown, so the spike proteins that surround the coronavirus, and then structures have also been solved of this spike protein in complex with the receptor that's found in human cells. Using drug discovery is slightly difficult because we need a very high resolution structure to do drug discovery. So even though preliminary structures have already been found, I think a lot of them are not high enough resolution. So that means that we can't see the small atomic details yet. to form to sort of synthesize an effective drug that can bind. And I think another problem is the length of clinical trials. So even if we can, so the most effective way to use structural biology right now would be to repurpose an existing drug. So maybe use a drug that's already been approved by health agencies like the FDA in the US to use for curing COVID. And as we know, we don't know of such a drug yet, right? So that means that we probably have to synthesize something novel that can specifically bind to the coronavirus. And that means that it has to go through clinical trials, which, as we know, takes months to years. So that's probably why structural biology is still lagging in trying to find a cure for this disease. A vaccine, on the other hand, is like a completely different approach.
Actually, we do have another question in the chat. Do you use cryogenic chamber to crystallize protein?
That's a good question. So the protein crystallization process itself has to be has to go through trial and error. So we usually do the crystallization process either at room temperature or at 4 degrees Celsius. Once we do get a protein crystal, we actually have to plunge freeze it in liquid nitrogen. And as mentioned earlier, liquid nitrogen is like at minus 180 degrees Celsius, 300 something Fahrenheit. So that's like a low temperature. And that's usually done in order to preserve the crystal, because when we use it in X-ray diffraction experiments, the X-rays are very high intensity and they can burn the crystals in a fraction of a second. So the low temperature helps to conserve the crystallized protein. But when we're crystallizing the protein, it's done at room temperature or at 4 degrees.
All right. And the last question is, how does protein intake associated with immunity?
So, I'm not sure how to answer that question. So immunity, so as we know, so antibodies are actually proteins. And so I guess proteins form a very important part of our immune system. I'm not sure how to answer protein intake associated with immunity. So there are certain proteins. So for example, the spike protein in the coronavirus crown is a protein that specifically targets a receptor protein that's found on the surface of human cells. So it's this protein-protein interaction that actually guides the virus to a specific host cell, which is the human cell in this case. So I guess proteins, so that's an example of a protein that can be deadly to us. But an antibody, on the other hand, is a protein that our immune system produces that can specifically identify pathogens, target them for killing them. So I guess proteins can be both good and bad.
All right. Thank you so much, Kafrik, for all your answers. And let's go to Kahoot right now to respond to the question that we have.
I think this time I should be the first one to answer. No, not yet. Okay.
All right. Thank you so much, guys, for your answers. So right now, we'll go through our third presentation, and it's from Lejla Dervisevic. She's a fourth-year PhD student at Purdue University studying international relations and environmental policy. She researches the role of non-governmental organizations, NGOs, and corporations and global environmental governments. And when she has some free time, she enjoys dancing, watching movies, and cooking. And today she's gonna be presenting about the role of NGOs and corporations. So we have Lejla here with us. She's gonna be presenting live. So she has a presentation ready. So any questions, guys, you can just shut it at the chat. So Alex, I don't know if you can just share the presentation.
Hi, I hope everyone can hear me. I have some difficulties with my technology this morning, so hopefully everything is going to go well. As you already know, I'm Leila and I study political science. So my lab, I don't have a lab actually. So we like to say in political science that the world is our lab. And so there will be no really cool animals or insects or lab pictures in my presentation. But I do study how we address environmental issues. So how does our society address problems related to the environment? So some of the presenters before me talked about, for example, how pesticides affect different species or how we decide which drug is approved or not. So my part of research or my job is to study how we get to decide what amount of pesticide is good enough, or how do we get a regulation that tells us that we shouldn't spray pesticides around a lake or a body of water. So I specifically study how corporations and non-governmental organizations participate in this process of making decisions about environmental problems. So if we could go to the next slide, that would be great. So I will just briefly mention that not always were we worried about environmental problems, and not always did we ask our governments to do something about these problems. So the moment when we started being worried was when we actually realized that industrialization or really fast development around the world was doing terrible things to the environment around us, and we could actually see it. So in the 1960s, specifically in the United States, we saw an environmental movement and a lot of activism at both individual and local levels. I don't know if some of you heard about Rachel Carson and her book called The Silent Spring, where she writes about the fact that springs in the United States are becoming very quiet and silent because birds are dying as we spray pesticides all around us. So at this time, we're also asking our government to do something about problems such as pesticide use and their effects on human health and human environment, as well as air pollution and water pollution around us. So we're seeing in the United States how we're forming new agencies such as Environmental Protection Agency, and also enacting policies and acts such as National Environmental Policy Act. Next slide, please. But we're also around this time realizing how many environmental problems are actually not contained within boundaries of a certain country. So we're seeing that although they're produced locally and caused locally, for example, climate change, we are emitting CO2 and other greenhouse gases, sometimes at an individual level by driving our cars, or at the firm and company level. But the effects of these problems are very global, meaning that everyone is affected. We can't keep CO2 emissions within the United States, but these problems cross boundaries. So we're also then seeing that we need governance at a global level. So we need to address problems not only within countries, but across countries at a global level. So next slide. I think this one will just list everything I talked about. So what we're seeing at this time is that all these governments or governmental leaders that represent different countries come together in order to address problems at a global level. And what I study is global environmental governance, and how we define governance in political science is that it is a sum of policy instruments, mechanisms, rules, norms, and procedures that we use to identify problems and then address them at the global level. And what we see at the beginning is decades of state representatives and state governments coming together and negotiating at the UN and coming up with treaties to address these problems at a global level. So there are conferences happening from 1972 in Stockholm to maybe all of you are aware of what happened in Paris in 2015. Next slide, please. But what is interesting in this process of governance is that recently we've seen that not only state governments are making rules, but private actors are also being engaged in addressing environmental problems. So we see non-governmental organizations being more engaged because they monitor what countries do. They write reports about how countries are behaving or misbehaving in terms of environmental, their environmental agenda. They also decide which problems matter, and they lobby and negotiate at the UN now. They're part of UN negotiations, but we're also seeing corporations driven by the corporate social responsibility idea starting to self-regulate. So they make their own rules about how to make their supply chains more sustainable, or they're creating private institutions where they come together as corporations and decide what product is certified as produced sustainably, or what is organic, or what is GMO free. So we're seeing what in political science we call emergence of private governance, and it's somewhat replacing what states do. So it's replacing our governments because governments have been failing to address the problem. So we're seeing private actors coming into play and actually replacing governments. Next slide. So I personally study partnerships between non-governmental organizations and corporations. So we're seeing more and more how environmental non-governmental organizations such as Greenpeace are working with corporations in order to address problems related to our environment. So I study them in terms of what they look like and what their effects are on global governance. Are they effective? Are they doing a better job than our governments? But I'm also looking at the consequences of this phenomena. So although they may have more capacity to address some problems, we have to understand that we don't really enact or vote for corporates board of directors, or even NGOs are not really elected. So in terms of governments making decisions on behalf of us, we have some form of or mechanism to keep them accountable because we vote for them, or we can vote them out in democracies and keep them doing a responsible job at protecting us. But when it comes to corporations taking care of our environmental problems, we don't really have any way of keeping them accountable in a political way. So that is basically where my research is at, and I'm kind of still gathering my data and writing my dissertation. So I would be happy to answer any questions.
Hi, Lala. Thank you so much for this, wonderful presentation. And it is always good to, look at the dimensions other from what you are working on. So it has always been enlightening whenever I hear you and your insights into all the work that you do. So I had like two questions when I was going through your video. How much scale matters? Like if there is anything that is going on local scale, and any challenges, environmental challenges, and in NGO working upon those local scales. So how much time does it take to highlight those things at UN or make this a global thing? Because as we know, fractal, whatever happens in one city also happens in other city. So can you share some insight on that? Like how much time does it take, or is it really strugglesome to take things from local scale to global scale?
Thank you, Alka, for your comments and your question. So I actually just recently, so one of my minors is ecological sciences and engineering, and I just gave a similar presentation to a group of students there, and we talked about scale a lot in terms of, and there is a big set of literature talking about if problems are caused locally, shouldn't we then address them locally as well? Because then if we do that at the local level and we engage local actors, at that scale, we have more knowledge, we have more local knowledge. We can engage communities that are actually experiencing these problems to tell us what they need and how they want this problem to be addressed. So there is this term called polycentric governance that says we shouldn't have a central decision maker, but we should have communities address the problem. So we're kind of moving away exactly because of what you mentioned, that it's really hard to, at a global level, really portray what's happening locally, given that it's happening all over the world. And there is a cultural component to that. There is identity component to different communities wanting to govern their resources differently. So we are moving somewhat away from doing the centralized governance at the international level to doing it more locally, where communities are basically deciding how to do it for themselves.
Okay, wonderful. Thank you. And my other question was, which I forgot kind of, I thought you answered that in between. So I think we can take somebody else's question. So Anush asking, should we really depend on government to act? Yeah, I mean, so I think that was somewhere linked to that. Whenever NGOs make any, act or work on something, how difficult it is to get acknowledged by the government, and as you had mentioned in your last slide, the consequences, the accountability. So it's really hard that government appreciate those NGOs effort, or what is the percentage? Do you really see clashes between NGOs and governments act, or is a coherence in between?
So in terms of relying on governments, a lot of people are saying, no, we shouldn't, because they've had decades since 1970s to address the problem, and they haven't really effectively done so. And thus, many people are okay with these private actors coming up and actually taking care of the problems. So in terms of the relationship between government and NGOs, or government and corporations, so I study private partnerships. So I study how NGOs and corporations work together, but there's actually more recently also a lot of public-private partnerships. So governments are realizing that they can't do everything and they don't have the expertise. And NGOs oftentimes have, first of all, local knowledge. So they're more engaged in the community, so they will know what's happening locally and will be better at implementing policies at the local level, because they understand the dynamics of the community. But they also have experts who are studying these issues, and they can actually share that with governments. Same with corporations. Many corporations actually know better their supply chains or what they can do along their supply chain. They have the money and the capacity to do it. So governments are actually now cooperating more with NGOs and corporations. So we see a lot of that.
Wonderful. Thank you, Laila. Sanchez.
All right, thank you, Leila. I think I do have a question as well, because the problem sometimes with NGOs is that the poorest countries are the one with more NGOs, and sometimes you see that it's just something they're doing to showing off instead of kind of really targeting to the real problems people are facing. If we look at an example in Africa, we have a lot of NGOs, and sometimes they're doing nothing. They're just doing great reports. They're just doing big events. But when it comes to kind of going to the substance of the issues, they don't really show up for that. So, what is your take on those problems?
So there's a lot of research done on NGOs and whether they make a difference. And I think that what we sometimes forget is that because they're a nonprofit organization, or because we see them as acting on behalf of us as civil society, we sometimes forget that they're also have instrumental needs and goals. So they also want to have money and get funding. And so sometimes they actually forget their purpose and forget to do what they initially were formed to do, because they're running after getting money and getting support. So that's what makes them sometimes ineffective, especially when we see that a lot of funding comes from the Western countries and developed countries, or that even these NGOs were formed, or their headquarters, for example, Greenpeace is a Western NGO, although it has its offices all over the world. Its funding and its main kind of life support comes from the West. So very often, even though they are placed locally, they don't really understand that there is this idea that they impose this neoliberal way of doing things. So although we think, oh, they understand what's happening locally, very often they're just imposing the same agenda like the governments would. So there's a lot of criticism about NGOs that are the largest ones and most successful ones are usually the Western ones. So I would say that there is a difference between more local and actually authentically local NGOs that were formed bottom up, right? In comparison to top-down NGOs. So there is literature on both, and some of the bottom-up ones are more effective in terms of knowing what's happening, but they don't have the resources very often to act. So there is that whole thing of resource versus purpose, right?
Yeah. All right. And we have the last question. Does environmental injustice play into your research?
Yes, so that's a topic that cannot kind of be ignored, no matter what you're doing in terms of environment. I do not necessarily I have colleagues who study specifically environmental justice related issues in the United States and more globally. But a big part of, so my research, for example, talks about the fact that this division between global South and global North, meaning developed versus developing countries, has always been a part of a problem. Where developed countries are causing more problems. They are the ones contributing to climate change more than developing countries, but they're not really doing much to address it, or they're expecting that everyone should now come together and address the problem that they created. So there's a lot of injustice in terms of expecting developing nations to do more. or sometimes they don't even don't have the capacity to do it. And many developed countries don't want to help them. So there is that problem, but also in terms of corporations, when we talk about who is making decisions, it's again, very often multinational corporations that are headquartered in the United States, and they're obviously going to prioritize their own profit. So it's possible that people who are not wealthy and people who don't profit from corporations or don't have shares and that they will be most affected or be ignored in decision making. So there is that in my research, that's how I kind of talk about environmental justice. But there's definitely people who talk about it in more detail in their research than I do.
Thank you so much, Lejla. That was such a great presentation. Thank you for all your
You're welcome.
information and answers. So sadly, the question for this session will be at the end because right now we have another presentation about LGBTQ, but the question for your session will be at the end. So we'll go ahead right now to present the LGBTQ Center, Lesbian, Gay, Bisexual, Transgender, and Queer Center. Yeah, kind of hard. All right, thank you. So right now we'll be presenting the video about LGBTQ Center. So Alka, I don't know if you can please go ahead.
Purdue is ranked nationally in the top 20 for LGBTQ-friendly schools and #1 in the state of Indiana. And a big reason for that is Purdue's Lesbian, Gay, Bisexual, Transgender, and Queer Center. As you enter, you're welcomed by a large study space and multi-purpose room, where every Friday during the academic year, the center hosts Fabulous Fridays. The LGBTQ Center is a dedicated safe zone that provides an inclusive and affirming location on campus for all.
One of the signature programs of the LGBTQ Center are the trainings and workshops that we do. You may know them as Safe Zone or Trans Inclusion Training. These are opportunities for the entire campus community. Global education is critical, and we try and emphasize the understanding of LGBTQ lives and histories through a global window. And so we take students on a 6-week, 6-credit-hour study abroad opportunity.
Off the main lounge, you'll find meeting rooms and a university-supported computer lab. The center also maintains an extensive library and displays a collection of original artwork and artifacts that communicates an all too often hidden history. With over 9,000 visits a year, the Lesbian, Gay, Bisexual, Transgender, and Queer Center has become a popular destination not only for the LGBTQ community, but for the entire Purdue campus. The LGBTQ Center is here to support and help you. Please visit us today.
All right, so just so you know, this is the LGBTQ Center, one of the most supportive centers we have here on campus. I mean, regardless of your sexual orientation, we just make sure to support each other and try that you're not being discriminated by any means. So that's one of the point of this video. So can we go ahead with the Kahoot question?
You can still join if you are not in there. So join it. I think, Sanchez, can you post that pin in the chat box?
Yes, absolutely.
I just posted it again. So it's in the
Okay.
chat at the bottom if anyone wants.
Yeah, so go to kahoot.it and then enter this pin 4740122 and play the game.
Should I go ahead and start?
Yeah, please, Alex. At least this time I should be the first one. Can you imagine? I've been struggling since last game when I'm going to be on the first. but I'm wrong. I did not read all the options.
Yeah.
Wonderful. VT. I think this is the second time you're on the top.
Yeah.
And pull your socks up.
Okay. I think we do have another question as well from Legisla's presentation.
What is the question?
No, I think the Kahoot from Legisla's
Oh,
presentation. Yeah.
so I actually have that question after the reactor presentation.
Okay.
Sorry.
And actually, I think we do have a general question here, guys. I mean, how to choose from Anisha. Thank you, Anisha, for this question. How to choose a research topic. It is just a general question. So I think a lot of you guys can go ahead and have your own answers. You're in grad school, so I don't know if Alka, Hannah, or even Lejla has like any answers for Anisha about how to choose a research topic. Hannah, do you have a thought on that?
Yeah, I mean, I would just say, I did research as an undergrad actually, and kind of how I got into research and how I decided what to do was I just met with some of the professors whose classes that I had, met with them, talked with them about what their lab does, and then from there kind of decided what lab was the most interesting to me as far as what they were studying. And then again, worked with the professor to see, she kind of asked, well, what questions do you have about this topic? And from there, I developed my research question.
Yeah. So just to add some other details, Anisha, it just comes from like curiosity, out of curiosity, right? When you a student, you have curiosity about some topics, so some questions come through your mind. Why do certain people act that way? Why this thing is this way? Why do people do that and do not do that? What this is happening? Why is it happening? So all those questions come through your mind, and you're trying to find kind of answers for them. So what you do is first looking for the area of your interest, try to identify what professors are really related to these area that is on your interest, and try to frame a research topic. And as long as you're making previous research about other people who have maybe made the same research as you, and as you're making all this literature review, and you'll be able to frame your research topic. So it's a process. It's not like a one-time thing. It's a process. You have to go step by step, but it comes first from an idea, from a question, from a thought that you may have about something, and you say you're gonna be investigating about this, about that. All right.
Could I add something to that?
Yeah, absolutely. Please.
Yeah, I just wanted to mention that, it's okay, especially early in careers, and even, later on in our careers a little bit further on, but especially early in careers, it's okay to change research topics too. If you start something that you think you're initially interested in, and then you realize you are not interested in that anymore, when you get into the nuts and bolts of what it actually is, it's definitely okay to change and make a switch and try new things, especially, like I said, early in career, it's time to try new things.
Yeah, most definitely. You'll have a lot of bounces. You think that you were interested in this, and when you get to it, and you see the challenges, the pain that comes from having to make that decision. So you'll definitely want to switch sometime, but it's a long step, but it's definitely worth it, Anisha. So give yourself some time, give yourself patience, be patient with yourself about what you really want to do, and it does take some time. All right. So if we do not have any further thoughts on this, thank you guys. I think that's the end of the fourth session. So Alka, you can take over. Thank you guys.
Thank you, Sanchez. I will again start by thanking everybody, however overwhelming it can be. But honestly, thank you to all the students who have joined in here. And my deep heart thanks to all the leads and the speakers. And as I am saying again and again, that uh, it is just, pinch of salt that you're seeing. There are plethora of ideas that is running around, and what we were just discussing when Anisha has asked, I honestly feel just be aware, look at the resources, be into it, and you'll figure out what you want to do research with. Because sometimes if you do not know anything, then maybe ideas don't come up. But when you start knowing things, when you be in it, and especially communicate, talks, for example, this platform that you have got, just talk over here. You have, experts from each field. They can guide you in certain or the other way. So I really feel communication is the key. Communicate, look for resources, and you can definitely come up with your research topic. However, there is one nuclear reactor lab that we have at Purdue. I would like to showcase that lab tour. I know it is strange because, we used to go there in person. We will be looking at our campus in person, but seeing all of them virtually is kind of, I don't know, weird, but it is like, yeah, we have to do it this way. So I just wanted to make best use of the opportunity that we have. So I will showcase you one of our nuclear reactor labs that we have at Purdue.
This is Purdue University's reactor number 1. It is Indiana's first and only nuclear reactor, originally built in 1962. For its first 54 years, it was licensed at 1 kilowatt, but will move to 10 kilowatt operation during 2017. The reactor is a central piece to the research and teaching mission of the School of Nuclear Engineering at Purdue. The reactor sits at the bottom of a 17-foot deep pool. The water makes the reactor run, provides coolant for the hot fuel, and shields operators from the radiation. The reactor runs on a plate-type fuel, which is a uranium silicide matrix encased in aluminum. The fuel plates are about 60 centimeters or 2 feet tall, a couple inches across, and are placed together in assemblies. This is an older demonstration assembly. The assemblies are filled with fuel plates and placed at the bottom of the pool in the reactor core. The reactor is officially classified as a materials test reactor, which means its purpose is to provide benchmark testing for candidate materials to be used in industrial settings. Today, we continue to use it to test materials, perform basic research, and educate Purdue University undergraduate and graduate students, as well as the public, about nuclear energy. These are our original instrumentation racks, which monitor the power level and period of the reactor. The reactor was built by the Lockheed Nuclear Corporation in 1962 and features state-of-the-art technology of the time. Yes, that includes vacuum tubes. The operator console provides all the information the operator needs for the safe operation of PUR-1, including the annunciators, rod height, power level, time, and emergency numbers to call. The annunciator panels give information to the operators of the status of various parts of the system. If there is a reactor shutdown, one or more of the tiles will indicate the cause of the shutdown. When the reactor is starting up, operators reference channel 1 to see the counts from a special detector called a fission chamber. The fission chamber shows from 1 to 10,000 counts per second. It also feeds a low-power period circuit. Unfortunately, looking at dials and meters only gives an instantaneous measurement. Sometimes operators need to see how a value has changed over time, so they use the chart recorders shown here for channel 1. The red line shows us coming down from high power after shutting down. The reactor is started by turning on magnets, which lift the control rods out of the core using 3 different screw drives. The magnet power is carefully controlled and managed with this box. The current, and therefore force of attraction of the magnets, is reduced at higher powers. When the power gets too high, the magnets are no longer able to hold the rods up. When they fall into the core, the reactor shuts down. These dials provide additional indication of the reactor's power and the period. As the value increases, a contact switch can be closed and the reactor will shut down. Multiple instruments can shut off the reactor for any number of the important things we measure. This is the reactor's coolant processing system. Our water chemistry is very carefully maintained to ensure the integrity of the components submerged in the pool. In fact, our facility could operate continuously for over 150 years with the current amount of fuel. Our lifetime is dictated by the degradation from the water. The pH and conductivity of the water must meet regulatory requirements and is checked weekly. Water passes through a deionizing cartridge, a demineralizer, as well as a heat exchanger, which is shell and tube type and helps us keep the water at lower temperatures. Radiation monitors are placed around the facility to ensure dose levels remain safe. We rarely see elevated doses, and an alarm will sound if doses rise too high. Radiation levels are also shown on the instrument racks so that the operators can see the current level without having to leave their post at the console. Anything that goes near the reactor core gets mapped on this plate. Here, you can see the ones with a letter and a number show fuel. SS1, SS2, and RR are the control rods. G and I are for the graphite reflector, and S is for our startup source. There is a lot of maintenance to be done at the reactor. We keep detailed logs to make sure we stay on time, on track, and on target. Our operators are licensed by the Nuclear Regulatory Commission. Most of the time, PUR1 has 3 licensed operators. We are very excited about the future of the PUR1 reactor. In 2016, a new all-digital instrumentation and control system was purchased. Designed by Mirion Technologies and Scientech Corporation, the new instrumentation and controls will enable the staff to take high-fidelity data and allow many more teaching tools for the Nuclear Engineering Department. The new system must be licensed by the Nuclear Regulatory Commission and will allow Purdue to produce elite engineers and perform important research for many years to come.
Yep. So guys, this was the very elaborate peek into our nuclear reactor lab. And I would like to add that the facilities have been, renewed, and there are new lab setups and new equipments that are working right now. So if you're really interested, you can go to Nuclear Reactor Lab Purdue page, just Google it and you'll figure it out. And then you can go through a virtual tour. It is also available there. So you can definitely do that. Yes. So do we have any more games to play, or we can proceed? Okay, there's one more.
Yep. So we have just a few more questions left, plus one from Layla's presentation. So we'll go ahead and I'll wait like a few seconds just to make sure everyone can get their Kahoot ready.
Yeah. So again, you can join by kahoot.it, and I think the ID is 4740122. I think we are a little bit off time, but I appreciate all the interactions and everything.
Sure. In that case, I'll go ahead and continue. Hopefully everyone is ready.
Mm-hmm. Let's see who gets this right. Nobody got it right? That's funny. This means no one was attentive to it. Anyway. There's on how long can... Most of the people that no again this, oh no, what you guys have been doing. Nobody got it right. But this is huge. For 150 years, they have the view. Wow.
Okay, so this one's about Layla's presentation.
Okay, how do we address problems at global level? They just talk. They negotiate and create treaties. They address problems only within their own country. I got it right, finally. Many people got it right. Cool.
This one's going to be a tough one.
Have you enjoyed NGS? I have absolutely. I don't know about anyone else. Yes, 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 yes. You have no other option. See, all answers are the correct answers. Wonderful. I think they have chosen their favorite colors. Congratulations, everyone. Congratulations, Spencer. And VTJ, whoever it is. Great. Hannah and VTJ, you guys are the winners. Expect something. Okay, so let's go ahead and wrap up the whole session. This has really been enjoyable for me and, learning for me precisely, I would say, and especially in this COVID time when there was 0 to 1% interaction in my life, this has been huge. I'll just give you some layout first of all. Let me share the screen. Whatever we talked right now over here, all these all of those videos are uploaded on the YouTube channel. If you look for Next Generation Scholars 2020, you will have all of the videos and presentations.
One of the highest powered nuclear research reactors in the US.
Fine. So you can go through all of the videos and the slides if you want to look at them again. You can also check our website, purduegradstudent.com/NGS, or just Google NGS Purdue. You will get on this page. This page will be updated. For now, it was just about the events, the gallery and everything. So we will update the links and everything over here also. So you can use these resources. This was for the students. Secondly, if you have any question, post our session. So you can always email pgsg.community@gmail.com. I think I have all the spelling correct. Sanchez, if you're saying something, you are on mute. Sanchez, you are on mute. Okay, I don't know what's going on. So if you have any question, you can, it's a little bit distracting though. So you can always email us at pgsg.community. If you know whom you want to address your question to, you can also mention the name of the speakers. However, after their permission, we can post their email IDs, but I don't think because of some security and all those concerns, it might not be. So you can always direct your questions to pgsg.community@gmail. So there are two things that I would like to end this session with. Now the floor is open to the students, however it was for the whole session, but still I would really like to hear any students' feedback or, whatever you learned or your interest, just share anything about yourself or anything. So any student can just unmute yourself and just talk. The floor is yours.
Thank you so much, ma'am, for this amazing session. I really enjoyed it.
Wonderful, wonderful. So is there any one research that you got really ideas from, or all of them were as enlightening for you? I think I lost you, Anushka, or you can hear me. Okay, I think I lost her. Anyone else? Okay, she's here. Fine. You have to unmute yourself, I believe.
Thank you, ma'am. We are glad to have this session with you.
Okay, wonderful. So of course, like more than thanks, is there anything, any one particular research, or you have anything in your mind, or just out of research, how is your school life going, or anything you would like to share? If you guys are speaking, we can't hear you. Okay. I don't know, something is wrong, but I think we will share a feedback form with you all so you can write whatever you want. I would really appreciate if any of you speakers who is available right now with us and any of us leads uh, would like to share what was it like when you were in high school and where you have landed up. Did you had any idea about this, that you are going to be doing this when you were in high school? Or it's something that you explore throughout your journey? Any of the leads or the graduate student can volunteer over here.
So honestly, for me personally, I didn't think I was going to be going to grad school. I didn't even know, like, consider it as an option when I was in high school. I like what I wanted to do then is like so different than what I'm doing now. Like back then I wanted to do like either architecture or aerospace engineering, and now I'm doing computer science. So it's pretty different. Like things can change, like what you're interested in can change, and like just have an open mind and like listen to people's experiences, see what they have to say, and then just follow your interest.
True, I completely agree. Anyone else, any other graduate student would like to share their experience from high school till here, where you are right now?
I just highlight what Alex just said. I changed even after like my second year of undergrad what I wanted to do. So don't be afraid, if you start thinking this isn't what I like, don't be afraid to change, even if you're 2-3 years into your studies. It's okay. People change their mind.
For me, I would like to share when I was in my high school, definitely I was very active in name a competition and I'm there. Be it science quiz, English quiz, dance, anything. So I don't know, but I had no idea that I'll be doing grad school. Even after my undergrad, I was like, I don't know, maybe I can go for any administrative services or anything. But one thing I would stick to was I should enjoy whatever I'm doing. And I didn't feel that when I went for job after my undergrad. It was like hardly I worked there for 14 days or something, and I decided this is not something I want to do. So the whole focus for me was whatever I have studied. I was not those muggers like who can just memorize everything and things like that. For me, I always used to say this should have to be in my DNA, so I should understand what I'm studying. That was one of my key thing when I was in high school. And that is how, like, few of the basic physics fundamental and basic mathematics, I feel, if we have, for me personally, I felt when I had the clarity about these two, the physics and the mathematics, I started seeing the world around me in that form, be it when I am in nature or be it when I'm doing my research. So I always associated with those fundamental laws of physics and those basic equations. So for me, it was what's something, just for example, a leaf falling off a tree. And that was my observation. And then I decided, okay, why is it falling like that? And whatever questions come to my mind, and think about any of the physics thing that is available right now to understand it, and then to comprehend it more, write an equation about it, and then solve it. That was my way of going about it when I was doing my master's. So at that time, I think I was watching rivers a lot, and I love Himalayas and Ganges. So I was looking into that. But after that, I just got very curious in hurricanes and everything. So presently I work with NASA, tracking all these hurricanes and how much damage it is doing to us. So that is my experience. So I think you just have to be open as a high schooler, because we study a lot in high school and we never know which is that one topic that I am very much attached to. And I really feel in your research, you have to be self-motivated. And for self motivation, I really feel something that you really care about in your society. Because as scientists, as researchers, or even as a human being, I feel the whole deal is what you are giving back to the society you're living in. So, yeah, just choose something. And it's quite interesting, I feel. Just choose something and play around that concept. And I would like to add something that um, Web of Science and Google Scholar is something where you can always look at your ideas. Suppose you have an idea about creating a tunnel. So go to the Web of Science or Google Scholar, just type your idea over there and you can figure out a lot of research and papers that has worked into that field, and you can explore it more. So yeah, I think that's all from me. Anyone else would like to say anything? Anyone, like students or the speakers or the leads? No? Okay, wonderful, guys. Thank you. Thank you so much once again. I know we have gone totally beyond time. We had restricted it for 2 hours, but now we are in 3 hour zone. I'll follow up with your emails and everything. Spencer, you were saying something?
I was just going to point out it
Thank
looks
you.
like there's a question for you in the chat.
Okay, I was not reading. What is the question? Please share us your steps from Raj Kamal to wonderful field of research. I think I just did. I can follow your footsteps. No, don't follow my footsteps. No, you should never follow anybody's footsteps. I believe, yeah, definitely guidance is something that you should look into, and you should always reach out to your professors, your, alumni for that. And you should explore your own journey, because everybody's journey is not same, so you can never replicate anybody's footstep, I would say. But I would love to interact. Just email me at pgsg.community@gmail.com. And as I said, check our PGHD NGS website and I'll follow up with the emails. Thank you once again. Thank you so much for this wonderful session. Thank you, guys. Thank you so much. Yep. Bye, everyone. Have a good night and have a good day to all of you. Thank you.
Bye-bye.
Bye.
Bye, ma'am.
Bye.
Thank you.
Thank you. Thank you for joining. Bye. Okay. So thank you guys for leading. And I'll follow up with more with emails and Slack. Thank you so much. Thank you, Alex. Thank you, Hannah. Thank you, Shansis. And thank you, Spencer. Thank you, Angel. I think he's not here, but thank you, everyone. Thank you so much. Bye.
Thank you, Alka. Great job with organizing everything. This is went quite well.
Yeah, I feel this might have been useful to many of the students, at least to me, because most of these things I never knew. Yeah, that's wonderful. Thank
I guess I had a quick question. Was there going to be any follow-up with the local high schools or anything?
Yeah, but I don't know, for some reason local high schools had not signed up, so that's why we canceled the Friday one. But we have recorded this whole session, and we will send out this one.
Gotcha.